Good afternoon, folks. With a little technical delay, here we are. So what I'm going to do today is talk about my career as a hybridizer and some of the rules that I learned during these 55 years of hybridizing.
The test is at the end anyway.
Yeah. I wouldn't be here today without these four women who led me to my hybridizing career. Polly Bishop was my neighbor in Athol, and she's taught me how to hybridize when I was 9 years old. And so I'm sure she had no idea what she was starting. And then she passed me off to B. Warburton, who was, of course, a great mentor, and she got me so interested in genetics. I have a PhD in genetics now. Lynn Markham, of course, you all know here. She was the BB queen, and I went to her garden from 1964 on. It was, and I even discovered Hot Canary before she saw it. And then Jean Witt, I never met in person until I moved west, but she encouraged me in the MTB breeding when it was a very difficult field. And she was also a member of a genetics panel that I joined when I was a teenager. So I want to tell you about different kinds of crosses and how they're used. One type of cross is called the outcross. And this is where you combine genetically very different plants. So say you had a blue iris that had very good branching and a pink iris that didn't. You may want to try to combine those two to get something that had the branching of the blue and the color of the pink. Unfortunately, when you do those kind of combinations, the first generation is not a good outcome. They generally are pigs. And you have to have a lot of patience to work those lines further to get anything that's acceptable. Surprisingly enough, even there in the hotbed of iris hybridizing in Salem, most people are doing outcrosses all the time. They don't seem to do any kind of real line breeding. So they're combining diverse genetics and occasionally getting things. But of course, when you raise 40,000 seedlings, you're bound to get something good. Inbreeding is kind of the opposite process. It's when you take plants that are closely related and cross them with each other. And I had a very vivid example of this as a child. I went to Watkins iris field in New Hampshire, and when you looked at his new seedlings, there was no ugly ones in the bunch. In fact, I was standing next to Lynn, admiring them and saying, I wonder how they get such wonderful seedlings. And she snapped her head around and in typical Lynn fashion says, Kevin, they bred all the ugly out. She was good with that. You know, she could snap a line like anyone. And so what Watkins had were all blues and whites. So they really were narrowly focused on one objective. And because they raised lots of seedlings from a very limited range, they had progress every year. So you can, unlike animal breeding, where inbreeding would oftentimes get to animals that are not healthy, in plants we can of course just compost the bad ones and with no guilt. In fact, a good compost pile is a wonderful thing as a hybridizer. So what I'm going to take you now through is chronologically through the iris season in my garden, and then also show you all the other crazy things I play with in the second half of the talk. So the first group I'm going to talk about are the miniature dwarfs. These are plants up to 8 inches tall. And when I was in Mississippi, I started to collect some of Hager's MDBs that are crosses of tetraploid MTBs and pumila. And unlike many MDBs, these grow well in Mississippi. So when I got to Oregon, I decided, well, this would be a really good line to work on. Unfortunately, when I got to Oregon, Terry Aiken, Paul Black, and Thomas Johnson all had that same very good idea. So, but it was also good for me because I could take the seedlings from each of those guys' lines and combine them on my own. So this is Thomas Johnson's Alas, which is out of that Hager complex. And I used Alas a lot. And I took it, this first group you see here, across this to Terry Aikens things that are out of the same line. And this was one of the seedlings out of that first group of seedlings. So I already had things that were very well finished. This has been registered as Kung Fu Panda.
Of course.
Because it really, it's a little bit blacker than the picture here shows. And it has a nice red beard. And I love the white edge around the balls. And its sister is Little Jackpot, which has a little bit of the yellow, which I was surprised to see. It was the only one in the whole group that had a little bit of yellow in the standards. Now, when I was a kid in Massachusetts, Polly Bishop would tell me when I got seedlings that were good like this, you don't have to think, Kevin, you know what to do. You cross back to both parents and you cross all the good siblings with each other. And so that was an important rule that I learned really early on, because if a seedling had interesting characteristics, it meant that both parents must have it for the seedling to have it. And then about half of the siblings should have that same characteristics as well. And so I've just started the next generation of these. I'll bloom another couple hundred this next year. But here I combine some red beards now with just a little blue spot and a nice white flower. So as this is some of my rules, you may see these on the test. So your first impulse should be set to self, sieve, or back cross the best seedlings. And there's a proof of the pudding, the Watkins patch certainly, and these ones I just showed you. Here's some of the other MDBs that are popping up. I'm getting some really nice, very, very dark things. In fact, the one on the screen looks like it should be. The falls are very black. And out of that same group, because I cross MDB seedlings that had plicatas in them, I got some very dark plicatas as a result. Here's another one here. This is I've always been fascinated with green irises, and this is one that reminded me of the old green spot that I grew as a kid. But it's a very nice flower, I think. This one stays tiny. This is, I've been playing with a little bit with the oranges, and Ben Schreiner, who's the next generation takeover Shriners, is doing very good work with miniature dwarfs and standard dwarfs now. And so they raise about 10,000 dwarf seedlings each year at Shriners now too. And so I've been taking some of Ben's things and crossing them into related lines, and they've been giving some very pretty flowers. The standard dwarfs, of course, the next group higher up in height. And for years, I kind of ignored them because A, everyone else was breeding them, and B, I really wanted that flower on an MTB stock, not so much on an STB stock. But I'm in Oregon and there's a lot of people breeding STBs. So one of my other rules is to take what I think will be an important parent and then cross it to the yard. I call this like a saturation program, where you take one pollen and just work it to everybody and see what hits. And and this gives you families that all have one parent in common, so that when you see those seedlings bloom, you can cross the different families with each other. So you're still inbreeding, but you've got characteristics of more than just two plants in those progeny. So one I used was one called Guacamole. In fact, I saw it at one of the AIS conventions. It's one of Jane Ritchie's plants. It's normally when you get green in the dwarfs, it concentrates in the spot. But this one, the green was kind of all over the place. So I thought it might be fun to use. Although everyone warned me, the pedigree was so weird that I was asking for trouble. But I still did it anyway. And what came out of these is I crossed it to the yard again, and I got all kinds of things. A lot of them have green influences, and a lot of them have very pretty blue beards. And they ranged some kind of more muddy greens like this one here, but with some nice shape. This one more pastel. And I got some interesting patterns just out of the blue, whether this is due to some of the weird genes in guacamole or something else in the pedigree, but I love this kind of double halo pattern. And it gave reverse aminas too. That was the other thing. That was kind of a shock. So, and then maybe because guacamole's pedigree was all over the place and itself, and it's going to give all these things. So I've taken these various approaches now, crossed the reverse aminas with each other, crossed the greenest approaches with each other, and I should see those seedlings next spring. So Yogi Berra was also a great influence on my hybridizing. And one of his best quotes is, "You can observe a lot by watching." And in Salem, Oregon, I got lots to watch. So there's just thousands of, there's about 100,000 new tall bearded seedlings that bloom every year just in Salem. And so you can learn a lot. And so one year I was over at Mid-America and Tom Johnson was just blooming the first group of seedlings out of a plant he subsequently named Alea. And he had done outcrosses with that plant. And almost every group of the seedlings had 8 or 10 things you'd want to name. And so I said, you know, you don't have to be a fool to think, okay, I bet I could do the same. And so instead of taking it to outcrosses, what I did, I took it to some of Marquis Smith's standard dwarfs, which were related to Alea, rather than just crossing it to anything willy-nilly. And that proved to be really quite interesting. So I got all these very cool combinations now of both pattern and ground color. And the ground colors are especially interesting in that I got pinks, orange, and yellow ground colors. And I'll show you some of these seedlings. Every one was just pretty. In fact, I started numbering and then realized just keep them all, Kevin. You're going to want to see them all again. But some of these are just really striking. Yeah. I love when you get blue styles on a pink flower too, or a pinkish tending flower. And here's the yellow ones. The yellow ones are a surprise because they shouldn't appear. I mean, it's a tangerine base. You shouldn't get yellow appearing. So, and it happened in Tom's patch too. So this may be a yellow that's not converted to lycopene like some of the others. So I definitely have crossed these. Some of them are just wild. Here's another one here with a very, the standards have the pink, whereas the falls have the yellow. Amazing colors. This is one that's fun. This plant is a maniac. It hit 8 stalks one year from seed. And that's even good for Oregon. I know, don't you hate me? The other thing I've been fascinated with the standard dwarfs is these spot patterns. And I've always liked blue spots with a deeper spot on them. And it's really one of my favorite colors. And they're so showy in the garden. And it's hard to make an ugly one. Oh no, these are all just seedlings here. Yeah, yeah. Although the last one, I had a television crew come through last spring to shoot the dwarfs, and they all wanted that one. In fact, it's featured on the television interview. It rained a lot in Oregon last spring. In fact, there was only 3 days in the spring where it didn't rain, and so a lot of my pictures have raindrops. In fact, some things I never get to see a decent flower on, so I decided just to leave all these guys that got rained on and take another look this year. But there's some very pretty little flowers here. Yeah, this one is actually, if you look on the screen here, you can see it's really almost black. And then because these lines are related, the recessives fall out too. Here's where most of the purple left the flower, except for the spot, and you see what was lying underneath was kind of a yellow. Yeah, in fact, Tom Johnson had a placata called Chow Bella, and he was also getting wonderful seedlings from it. And so monkey see, monkey do. I took Chow Bella to some related things. Excuse me?
We're doing the same.
Oh, yeah, no, it's a marvelous plant. And I think there were about 60 seedlings from it that I bloomed last spring, and there wasn't a pig in the bunch, so. And it gave all sorts of shades, oftentimes with interesting multicolored beards and very fat forms. This is out of Terry Aiken introduced a plant called Fairy Fireworks, which blooms 3 sets of blooms. It starts with the miniature dwarfs, blooms again the end of the standard dwarfs, and blooms again with the TBs. And so it's a hard parent. I haven't got lots of seedlings from it, but what I got is really incredible. This is Fairy Fireworks by My Share, and this one also blooms the 3 sets of blooms, but it has a little more interesting placata pattern than Fairy Fireworks. In fact, Terry Aiken was really excited about this one himself. He wants to put this in his catalog, obviously. So the miniature talls, oh, if I could spell your, it'd be good, wasn't it? The miniature talls were a class that I played with as a child. And in those days, it was a pretty horrible class to work with. They were nearly sterile. And when you did get across, you got a lot of kind of dull pastels and nothing too exciting. And some people just hated them because they didn't look like a modern iris, but I always found them interesting. They had that sort of a wildflower look to them. And also, you cannot kill them. So that's one of my rules, though, is that numbers do count. And so you need lots of progeny to get a really, to understand the genetics of these plants, and also to get some good progeny out of the group. So the more progeny that you're able to raise, the greater your chances for success. So about 1970, I raised a group of about 300 MTBs. And when Bee Warburton came to my yard, she says, "This is the biggest patch of MTB seedlings ever." And she may be right, I don't know. Up to that point it was. Now I can raise 2,000 a year, no problem. So out of that patch was one I named Real Jazzy. And it was one that I wanted to use in further breeding because it had what Gene Witt called the fall flip. It had just a little bit of ruffle to the fall, and it gave it a little bit of a lilt to the flower that was often lacking in MTBs. And then also, I was starting again from scratch when I was in Oregon, so I just went ahead and crossed all the best MTBs with each other and just hoped for some really good things, especially in dark MTBs, which is, we don't have many good dark ones. And then also trying to improve the colors and patterns. This is Real Jazzy itself. I made it when I was 14, so it was 3 times runner-up for the Williamson White Award. I'm the Susan Lucci of the Williamson White. little Bluebeard came in runner-up 3 times and lost by a vote. Real Jazzy came in second 3 times and lost by a vote. So anyway, Susan has nothing on me. Oops, whose phone? Nobody's. Anyway, this is Real Jazzy, and I'll show you. These are some of what we're going to see now, or some of my other breeding line. This is one of Chad Harris's reds, and I've been trying to breed in these lines too. And unfortunately, when I crossed reds, they didn't come out red. This is Purple Petite, which is one I put out 2 years ago, and it is a maniac plant. In fact, Terry and Barbara just said that they had to throw away quite a bit of it because they couldn't give it all away. So anyway, it's a really neat flower, and you can see what happens there. The yellow, which is the underlying color of a red, fell out in most of the flower except for the little petal edges and around the halves. And so the purple color that would have made a red normally is only just purple. Still, it's a very pretty little thing, and it's perky. So this is the group now that derived from Real Jazzy. And this is one called Booyah. That'll be next year's introduction. It has some of the smoothest falls of any of these. But what's interesting to me is that the variegatas in the miniature talls show incredible variability. In fact, I'm seeing patterns in these diploid variegata miniature talls that I've never seen in a TV. This is one that bloomed this year that I really like. It's very, very almost black falls and an edge. So it's like Real Jazzy on steroids. There's a pattern, variegata pattern, or just whiskers of the variegata pattern on the halves here. Or it goes to veins. So you walk down a row of these seedlings and it's just amazing variety. Here's one a little bit more tan than the standards. A full kind of mocha look. And this is one I named Peanut Butter Cup, and it was introduced last year by the Aikens. And it's really floriferous. It about 12 buds on the stalk, and it has these pretty kind of two-tone peanut butter flowers. There's a story on this one. Steph happened to be in my yard when this particular seedling bloomed, and being childs of the 60s, we could only sing one song. Ring around the collar.
Ring around the collar. Oh, dirty.
So it looked like the old whisk dirty collar rings, and there it was. It's kind of an interesting pattern. Here's another picture of the flower here. It's really kind of an interesting look. It's something we haven't seen in a TV either, really. So another variegata. And I'm trying to get the variegatas darker and darker yet. This is one that's called Mocha Latte. It's has brown standards and fairly black falls. Okay, this is the part, it'll be on your test. So one of the ways we can create genetic diversity are called transposons. And what these are small bits of DNA that move into a gene and suppress the activity and then pop out and allow it to activate. And they can bounce from one gene to another. So Gene Witt said, Kevin, you need to work with these kind of strains and see where you can move these transposons in other strains. So the next group of seedlings are all transposons. So here's every place you see a streak of purple that there was a little transposon jumped out of its spot and allowed for a purple streak to occur. The bigger streaks, so when the transposon jumped early in development, and the small streaks, when it jumped out late in development. So you get just a small spot. The other thing that, let's see, I think this is, oh, here's another one here. So the other thing that the transposons can sometimes do is when they jump out, they pull a little bit of the gene that they're working on out. So this one, it's pulled some of the gene out so that the blue doesn't work quite as well on the standard as it does in the falls.
So Kevin, on those things, are they the spotted patterns like these are generally unstable? Is that
Correct.
in other words, it doesn't do the same in every flower in the clump? So what's that about?
Yes, so that's the problem with these guys. So no two flowers are going to be exactly alike with these guys. And I have these jumping now in different loci too. In fact, this is one here where you can see in the standards, I've got the gene that transposon has jumped now to one of the yellow loci. So I'm now getting streaks of yellow in the standards, and I'm getting streaks of purple in the falls. So the transposon is now in two different genes in that particular plant. So I took this plant this year and crossed it back to non-transposon things to see if I can segregate out just the yellow striping and the purple striping from each other. So I think could be fun. Yeah.
What is behind the Kaspersky?
Yes,
TVs, I call it watches.
broken color.
Broken color, yes, that's all the same game. Yes, yes. Yeah. Here's another one of these. Some of these are just really cute. And I don't think I'd like them in a big flower, but I like them in these little flowers. I think it'd be too garish in a TB, but in an MTB, they're just fine. Now,
Oh,
and
that's
here's one
cute.
where all the jumping was very late. So you tend to get ones that either jump early or jump late, and also ones that are very erratic. And so you get all of those various combinations of these transposons. I like this kind of a look. So it is jumping in other loci too. And here's I have it mixed with placata. And here's some of the other things that are appearing in these crosses. I don't know if they're beautiful or not, but certainly different things are happening. It's moving around, it's doing different things. Here's a more standard click here, and you can see the dots are more even and more regular. But we think that transposons might be involved in these sanded clicks too. I've always liked aminas, and this is a rather lovely one. I had a whole series of these with very nice form, and I'm trying to decide now. I think I numbered 6 or 7 out of this cross, and trying to decide who to name and who to not name. But they have pretty branching too, very nice in nice form. Here's the neglecta one. The standards aren't quite as white. I mentioned I was working with dark ones, and so several years ago, I made crosses between Rayo Sedentro and Chocolate Fountain, which are two of the nicest dark MTBs. Rayo Sedentro's has striping on the falls, and Chocolate Fountain doesn't. So the first generation, I kind of got something in between, you know, a little bit of veining, a little bit of solid. And when Phil Edinger saw these seedlings, he said, "You need another generation, Kevin." Yes, Phil. And the next generation was good. I got some now that had saturation now in a self flower. And I also had some that had more prominent or darker veins than Rayo Sedentro. So it worked both ways. So by combining those traits and then crossing the siblings, I was able to get both the vein pattern and the solid patterns better. Here's another one here that bloomed this season, very saturated falls. But then you get surprises when you when you inbreed, grandpa's recessives suddenly pop out. And so I was shocked at a whole row of seedlings of these because about a quarter of them were flat. I know some people don't like flat, but and I wasn't certainly breeding for it, but I won't throw it away either. Here's another one here. I know, aren't they fun? Oh, I got a raise out of Steph on that one. Yeah, I know. It's like, I don't know whether to like it or club it, you know?
Five beers.
I know, the beers.
Not 6.
Yeah, anyway, so inbreeding shows its ugly head once again here, and grandpa's flat characteristics pop out. Gene Witt left me a lot of things from Iris Reginae with lots of striping in the falls, and this is some of the work in that area. This is one, it's called And Stripes Forever, and it really does have And Stripes Forever. It's a very short plant. It's out of one of Gene's seedlings and one of mine. And this is striped pants, that pinstripe pants rather, that's also out of Gene's crosses mine. One of the other things Gene wanted me to do, and there was, you didn't argue with Gene. And so she said, Kevin, you need to make a better ice fairy. Ice fairy was out of class. And so this uses one of her seedlings that was in that direction, crossed with one of mine. And it actually is kind of an improved ice fairy. This is called Ice Shavings. So I think she'd approve. as I mentioned before, the miniature talls have a very narrow genetic base. There were like the 8 original MTBs that came from Williamson. And almost every MTB we have now can be traced back to those 8 Williamson plants. So one of the things that we needed to do was to take other diploid genes, other diploid parents, and cross them into the miniature talls. And they've been doing this for years, but mostly not with good luck, because when you cross the MTBs out to another group, you got things that were way out of class. They didn't look like MTBs anymore. So way back when they used this one tiny little pixie seedling of genes to shrink the heads of the big flowers. But now we have a much better one, the hybrid named Aster Girl, which involves Astracanica, one of B. Warburton's gifts to MTB breeding. So this is Elfin Etchings, which is a 2nd generation cross of a plant Phil Edinger bequeathed Steph and I both, called Mullinax butterfly. And it had a very interesting placata pattern. And I crossed it with Aster Girl and then sibbed those seedlings. And this one came out, it's very petite, almost Aster Girl size. But interestingly enough, the placata markings are both in yellow and violet. So this is another one I'm using in breeding to try to get, I would like to see a full yellow placata with no purple, which I think could be very cool. I like Plex a lot. This is another of these same things. Aster Girl crossed onto much bigger things, and the next generation giving all sorts of pretty flowers. Lots of speckles. I know you like speckles. Some of these are really quite pretty combinations of things too. And things that I haven't seen a TB look like that or like that. So anyway, in fact, even the TB people that come to the yard, especially Keith Keppel, who has seen every placata in the world, said, okay, that's different, Kevin. Another one of these speckly ones. I also like the more classically marked placatas. This reminded me of an old Belle Meade that, in terms of the pattern, very pretty flower and very nice shape. We're getting much better shape in the MTBs these days. This seedling appeared about 5 years ago, and it reminded me of a seedling that I inherited not inherited, but was given to by Dorothy Gill years and years ago. And it was one where she had brown standards and blue falls. And I thought, ooh, what a cool pattern. But she never named that seedling, and I had no luck crossing with it. So when this one appeared, I thought, oh, this could be fun to play with. So I started crossing with it. And now I'm getting some things with clearly brown standards and quite blue falls. In fact, this reminded me of brown lasso kind of shrunk down to an MTB. Another one of the rules. You might see this on your test. B actually taught me this rule, and that, excuse me? Whites are important, and they're kind of like blank slates. You can work with them. They're genetically very neutral in terms of their own color, but they also have, oftentimes other recessives are lurking there, and oftentimes they have quite good shape. So this is one that's one of that I use a lot. It's recessive both at the placata locus and the white locus. So I can cross it with almost anything and get that other. So I cross it with a blue, I'll get a blue. I cross it with a placata, I'll get a placata. So it's recessive at both of those traits. So it's like a blank slate for me in terms of breeding. It has wonderful shape, and it passes on both the shape and the bud count to its kids, all the much better. The border beards are a class I've been breeding with for years. I was inspired by Lynn Markham's garden early as a kid. And when I started breeding irises again, I started breeding border irises. And as she would tell you right here now, it's a damn tough nut to crack. And so, so I have two kind of lines I keep going. One of the border bearded lines that I use are just small segregants from the tall bearded lines. And then the other lines have a filler added to the stew that make for more vigorous plants and a little better spacing and bud count. And I use a lot of Lynn stuff, of course. This is out of Teapot Tempest. This has that look of Tempest, but more, this one's navy blue. And these are out of crossing Terry's Aikens line that go back to a cross of brown lasso by an orange years ago. I know I wouldn't have done that cross either. But anyway, 6 generations later, the clear oranges are coming out too. And some pretty yellows. This is one of my favorites. Unfortunately, it's resisted every chance I've had to cross with it, the little stinker. And I've taken Lynn's dark lines too. This has got 2 doses of simmer in it. And the simmer plants tend to be really good. In fact, it's more the color of the picture on my screen here, quite a bit darker than what you see on the screen there. And this is some of the latest things I've been doing is combining the Affilibred line with my small TB line. So this is some of the Terry Aiken line crossed with either Lynn's or Paul Black's things that are Affilibred. And this is just an example. I raised 47 seedlings out of this cross, and every one was a border iris. And that just never happens. And they were all actually pretty flowers, had pretty branching, and are vigorous. So even though I wasn't able to cross with them this year because it poured every day, there's plans for them next year. This is out of my Preppy line here. This is, again, combining the Preppy line with some of the Affilibred stuff. Unfortunately, I didn't get this flower when it's fully open because it rained shortly thereafter. So, but it shows, these are very tiny flowers on 24-inch stalks. So they'll undoubtedly stay in class. This is a sibling here, which I kind of like that just minimal pattern of placated cheeks on an otherwise white flower. And Preppy, I've been breeding with Preppy, and this is an inbred line from Preppy. Preppy had a grandparent that was horned. And after 3 generations, poof, there's a little horn on this one. So, and it's kind of cute on a border iris. I don't like them so much on TBs, but I don't hate it on a BB. It's actually kind of cute. Here's some more of the Affilibreds cross into the standard TB derived BBs. Another one here. This is a cross of some of the Craig IBs into BBs. This one is a very tiny. It's actually probably more an IB than a BB, but I love the little dollop of blue around the falls. And I pulled it down just because it was about to rain. So I had to take a picture quickly. So I live in Salem, Oregon. Yeah, it's a little out of whack there. There we go. I live in Salem, Oregon, and as I mentioned, there's 100,000 tall bearded iris seedlings that bloom every spring. And so I quickly learned that if I wanted to breed tall beardeds, I would mainly be banging my head into the wall trying to do that, and all I would end up with is a bruised head. And so I stay away from those now and do other things. Although there's one project that I've stayed with for quite a while, and I have this in my head in this project. It's a quote from Polly Bishop, which you would go through my seedlings. Not your best work, Kevin. When she would look disapprovingly at something I was enamored with. And so, but I think she would be impressed that I was able to take something that was fairly ugly and made it at least semi-okay. This is the plant I started with. It's called repartee, which has all kind of problems, but it has really red falls. And it's a recessive amina, unlike the dominant aminas that we mostly see. And it's a cross of a purple amina and a yellow amina. And so the yellow aminas, of course, have improved greatly since that time. So I took repartee to the best yellow aminas, and I got some very fun stuff. So anyway, I uncovered some of those transposons in these flowers too. And and you can see this is a much better form than repartee. Thank you, yellow aminas. But I also got them things that are pretty red. I mean, for a TB, that's damn red. And that one too. Keith didn't say the word impressed, but he was like, okay, that's red, Kevin. Anyway, they're fun. Some of them have little texture veins and things. And this is a cry. Sometimes I just can't help myself. There's two irises growing next to each other, and they're just begging to be crossed. And so I get things like this. You know, I'm a bad boy at times. Okay, this group you can all hate me for. I'm able to grow Pacific Coast natives. Of course, I'm in the Pacific, and it's really amazing. So one of the things I'm doing is working with the Valley Banner pattern. Valley Banner is an iris that what's her last name? Anyway, Ruth Hardy found in Eugene that had striping on the falls. And then I'm working just with that pattern. I'm also trying to combine that pattern with a yellow ground color. So they are just ugly as all get out.
For those that don't know, these irises do not grow here.
Yeah.
Don't forget how to attach to them.
So,
But anyway, you can see why I'm enamored by them, though. And you get every kind of a,
okay.
yeah.
All right.
Yeah, I know, it's horrible. I know. So the Valley Banner pattern, I get in all different combinations. Sometimes where the veins have a spot open and just the edges, and you get every combo thereof. It's just fun walking down the rows of these. So I wanted to combine it with yellows, and I had this quite good yellow seedling here that had a little bit of veining as well, which intrigued me that might be something to work with on these lines. And so I took this yellow seedling and crossed it with my best stripe seedling.
Oh, Jesus.
And I get all these, and so there's little combinations of yellow and white. This one has very little yellow, but I'm not complaining. And this one has lots of yellow, and I don't find that particularly attractive. It's okay. Or they go like that, where you get some variation on the Valley Banner pattern. And you also see those yellow veins now from that yellow parent now appearing there. And this one here, just amazing, you know, it's very saturated colors. This is one that I really like this spring. In fact, I got quite a few crosses with this one here, where you just had, you know, bits of streaks of the Valley Banner pattern, so it didn't seem quite as dirty, you know. This is my favorite one though. And you notice what I've got now, I've got the yellow veins from the yellow parent all the way down the falls, and they're being superimposed by the Valley Banner stripes. So that one I used a good bit as a cross too. So anyway, they are just so fun. So I'll never be able to compete with Marty and Jan in Siberians. So, but I wanted to do something because I just like them. And so one of the things I did was most of the so-called red Siberians have this peacock blue flash right at the signal. And for years, back when B. Warburton was doing the reds, doing initial studies of the reds, she was appalled that all the reds had that peacock blue flash. Let's get rid of this thing, Kevin, she said. No, B, I did just the opposite. So I started crossing to get that peacock blue flash bigger and bigger. And here it is here. Now, these are just a bunch of seedlings. where I've got peacock blue flashes in places other than the signal. And here's where the whole falls when peacock blue flash, and the standard stayed red mostly, or magenta. And here's where the falls are edged on one side. And these are interesting. It's the exposed part of the bud comes out red. The unexposed part doesn't. So there's some kind of a photogene thing going on there as well. Another one here, you can see the red, the magenta is just on the mostly on one side of the petal, not the other. But peacock blue is much bigger now. Another one here, almost all peacock blue. Another combination of magenta standards and peacock blue falls. And here's one where it's fairly cleanly separated and nicely formed. The other thing I'm playing with, oh, this is one, unfortunately this picture does not show it well. This is one where I have the nicest, fairly uniform edge. It didn't seem to go sided in this one particular flower, and I'm not sure why. It was the only one out of that whole group that didn't show any sidedness. So anyway, I self this one now, and I'll see what the next generation comes. I also crossed them into Marty and Jan's line, thinking, well, it may be fun to see what peacock blue moved around does on that yellow background of Marty and Jan. This is the first generation stuff here, and they pretty much look like Marty and Jan stuff. But so I've now crossed these back to my best peacock blue spreader and see what will happen. Another line that I'm playing with a little bit is blues with white styles. And when I was a kid, there were lots of these blue Siberians with white styles, like Summer Sky, and we don't see that pattern anymore. So I took one called Mandy Morse, which is probably a cross of Summer Sky and Tycoon, and crossed it into some more modern blues and whites. And I'm gradually getting things now that have at least nearly white styles and blue petals otherwise, and don't have quite as icky form as Summer Sky does. And I even have a Versi. As a kid, I used to go out and look at Iris versicolor, and I was on some of the trips that Bee made to Maine when she would scream to Frank, "Stop the car, Versi, Versi," just scream Versi. And so anyway, this is about a 4.5-inch flower, which is really big for a Versi, and it's a fairly good clear blue and has an ultimate ton of buds. So I may do something with this. Spurias, I know not many of you here grow them in New England, but they're in the West Coast. They're very easy. You just plant them and forget them. I started this line in Mississippi, and so most of this line started with one plant called Offering, which had really good shape, and I took Offering to the yard basically, and crossing Offering, Offering has incredibly good form. So, and it was like a blank slate. It was just a cream flower with very pastel color. So it was like a blank slate. So anything I took it to, I got the color of the other parent. And this is one called Band in Boston that just got an AM this year. And it also has a little bit of the ruffling and form of Offering, but now in a much more interesting color. And of course it's Band in Boston because I have that band of purple around the falls. I almost named it BAND in Boston, but and its seedlings have been very interesting. This is one here. I'm trying to get the center of the signal area into deeper shades, like this one here, and then more towards dark. And I'm starting to get now some really almost black and yellow bicolors. And they're very neat, I think. This is Angel's Smile, and this also involved Offering and some another roughly ones. And this has just tremendous amounts of buds, which not the Spurias don't have. And it's a little smaller plant, about 35 inches tall. And it's an incredible parent because it gives these roughly beautiful things all different shades, but blues and yellows and fat, like a fried egg there. Here's another one, fat and blue. And of course, reds and everything are a pain in irises because we don't have red as a natural color. This is the nearest I've gotten to red and spurious, but it's kind of an archaic looking form. And most people would say, well, it's not really red, Kevin, either. So, but back to the drawing board. You know, for 30 years, I grew Louisiana iris, but I got a rude surprise when I moved to Oregon that I could make crosses, but there would be like 2 seeds in the pod, and the 2 seeds would not germinate. So it's been frustrating. I maybe get 20, 30 seeds a year after 100 crosses. It's not the way to go. This is one of those. This is a key lime pie that our friends at Plants Nouveau are actually working on. This one here. It has the key lime styles. This is Linzer cookie, which is little, as you know, that Austrian cookie with the raspberry peeping out the edge there. This is Razor Edge, which is from that same line of breeding, but now yellow with a red edge. This is another one of my rules, and don't try to do everything. Be like the Watkins and breed blues and whites, you know, you'll get much further. I'm sorry. Vaughn does not follow Vaughn's rule here. In fact, my name is Kevin and I have a problem. Nothing with pollen is safe in my yard. So irises are really just the tip of the iceberg. In fact, I have major efforts in a lot of other crops. And almost everything with pollen in my yard has had a tag on it or time or two. And so I have about 200 varieties on the market and another 100 probably in production. So one of my projects was on the Sempervivum. I started when I was 9. I was riding my bike past Polly's house and she was out crossing, and I pulled in the driveway and she showed me what she was doing, and she said, "Come back tomorrow, Kevin, you can make crosses." Well, baby, I was there. And so anyway, in my first crosses, I got 3 plants that actually went onto the market. And I had 2 medals from the RHS when I was 21. So little did they know I was 12 when I made them. You know? They'd hear the plants. This is Lipstick, which is one of those early award winners. It's a really brilliant red velvet. And this is Jungle Shadows, which I named after the border bearded iris of those same colors. And Jungle Shadows, is as you see, has been a very important parent for me. It was the only, at the time, it was the only SEMP that retained its color all year long. In fact, when they voted the awards for these plants, when they came back in August, it was the only purple plant in a sea of green Sempervivum. And so this was the one that got the silver medal. So. Well, yeah, most of the time I'm smarter than a bee. Most of the so-called breeding of Sempervivum was people just collecting bee seed and rolling it out. In fact, Ed Strachey would grow 20,000 seedlings every year, and I would raise 200, and we have the same number of introductions. So the bees just mix things up. You don't know what's going on, whereas I could make a cross in a direction and get there. As I mentioned before, hybridizing is a numbers game. So the more you grow from a cross, the greater you have something good. And the hybridizer's most important tool is that shovel to pitch out the ugly ones. And people come to my yard just thinking, you are going to throw all those away? Yes, Kevin. Yes, people, I am. So here's like, I grow about 20,000 SEMP seedlings a year from hand crosses. And these are 2 of my colleagues from Michigan that are come to my yard to pick out new plants to be put on the market. And everywhere you see those little flags or something they decided they would like to sell. So that was quite good. So when I started breeding Semps again in Oregon, one of my goals was to make a black Semp and to make something the size of a football. And you'll see some of the latter work. This is some of the black work. This is patent leather shoes, and it actually has that same shine that patent leather shoes does. And it's actually quite black too. And I've gone even blacker. This one's a bit bigger here too, very wide.
Ooh.
And those transposons are everywhere. So this is one that Chris Hansen and I jokingly named Jackpot, because we know that everyone and his brother will want to grow this thing. So, but anyway, so this is the same kind of thing is going on here. And this year I took Jackpot finally bloomed for me, and I'm taking it to all kinds of different things in the yard. So I should now see this, where the transposon goes. It could be fun. One of the interesting things about Semps is that there's 50 species, and everyone will cross with each other, at least to give an F1, and most of them will give advanced generations. So you have the traits of 50 different species to play with. So you can go wild in terms of all the forms and everything else. Colors, like whenever, coppers here, like this one. Copper with the black tips. This is one of the roller types. This is one that gets totally black in the winter. Some fuzzy ones. This is one I just named crab legs. Obviously why. It's about an 8-inch Semp. It ain't the biggest one yet. When I was a kid, I made one called Stuffed Olive, but it never was exactly those shades. So I've been working to make one that was really olive and really pimento, and I'm getting there. I'm getting some things that are close. This is one I have a nickname of red licorice, and it is the first red I've had that stayed red all year long. And it goes back, of course, to Jungle Shadows about 4 generations before this. So it has some good genes. Getting interesting shapes too. Here's some where all the leaves are fat. And we're getting things like we see in other gems. This is what I call the granulosa mutations in Echeveria. And I'm getting them in the Semps now too, where you get these bulbous projections from the leaves. Here's one in red. I know. Do you want to stomp it out, Steph? You like it? Yeah, it's different. It's different. Mrs. Spaghetti with marinara. So because, as I said, all 50 species, you have lots of genes. So there's some genes for very narrow leaves in the population too. And so you combining those narrow leaf things with red things, now I get narrow and red. And you can tell this one is really prolific. Or fat. I get really fat leaves now too. This is about a 9-inch diameter. Or this is Mad Hatter. It's cristate. So the meristem instead of forming a dome, it forms a line. So the leaves come along a line rather than a dome. Another one of my rules is crossing pretty faces is a good thing. And if you don't know what you're doing, it's a good place to start. It's a nice thing too, in that oftentimes those crosses are very nice themselves. And so if you're doing a lot of experimental crosses like those red aminas, you may want to do a few of these other pretty crosses in order to get something that's nice. So this is, can you guess what these are? Daffodils. Yes, it's a little slow to get daffodils. Even in Oregon, it takes 4 years to get daffodils to bloom from seed. So oftentimes I forget why I made the cross by the time the thing blooms. So, but here's what the 2-year seedlings look like. And so actually this whole crop of about 500 will bloom next spring. So I'll see lots of daffs. And here's a seedling bed of daffs here you can see. And so I'm just going to run quickly through these so you can see all the different ugly daffodils I've created here. It's hard to make an ugly one, I found. I know, I have a lot of people come and look at one of my culls out of this group too. I like these little fringy things. I'm getting pretty good orange cuffs now on white. Yeah, this is the yellow pink. So as and you'll see there's a little bit of pink showing through on the petals of this one too, as well as the cup. And I have it more so. And yeah, here's one here. It's white with a pink coming under the petals just a bit. And I've crossed these all together, of course, to for the next generation. This is my Cheetos line. And you can tell where the perianth has gotten there sampling the Cheetos in the cup. So it's a little bit yellow now too. And it's weird too, the daffodil people hate this kind of bleeding over, but I think it's one of the coolest things ever. The Dutch like it, so I don't care, you know. Yeah, some of the pink ones are hard to be.
Is that pink? Really that pink?
It's really that pink, yeah. This is another pattern I like. Yeah, this is, it goes back to one of Grant Mitchell's called Daydream, about 5 generations before this. And I love that white cup on the lemon yellow flower. And of course you get these bold colors too. And I'm getting fringies on some of these lines. Little fringes. Even white fringes on yellow. Oh, here's one. This is the seedling where the pink is really spread into the perianth as well as the cup. And here's an interesting combination of that daydream pattern and some pink infusing into the cup, but the base of the cup is white. So I'm seeing combinations I wouldn't have expected. Sometimes I always rave about recessives, but sometimes dominants are a fun thing too. And one of the dominants I played with a lot is with the so-called split cup gene in daffodils. And it's, as a dominant, it's great because what you can do is cross a split cup with anything else and boom, you get split cups. So I'm going to show you what happens when you cross normal daffodils with split cups. You get all these amazing combinations of now split cup. And the split cup, a normal daffodil cup is composed of 6 petals that have fused to form that cup. What the split cup gene does, it says separate into the 6 parts. And it's interesting to me that how many different combinations you can get as to the extent of the split cup, how far it goes up the petal, how wavy it is. This is one where the petals kind of intercalate between the split cup. Really frilly cups, contrasty cups, flat cups, another flat. Ooh, yeah, it's that shade too. It's kind of flesh pink. It's like, yeah, I know. And here's yellow pink. I know you can all hate me. This one's a little overexposed, but you can get the idea. They're really roughly split cup. Excuse me, I can't hear.
How long does it take to introduce?
Oh, probably another 5 years, unfortunately, even for a small release. Although in Oregon they grow, I get about 8-to-1 on bulbs there. So pretty split. I even took the splits into the jonquilla types, and here's a multiple headed split. I don't like this one, but it shows you what happens when you cross a very small cup with a split. It splits into the 6 parts, but it's not enough. It looks too thin. You don't do that again, Kevin. Here's another one, just berserk. So Marty and Janet probably could write a book on this one. As a plant breeder, sometimes you have to select in increments. That is, you don't get what you want in stage 1, but if you keep working at it, eventually it comes. And this is a demonstration of that. So this is a jonquil hybrid, um, and it shows that daydream pattern of lemon yellow with a white area at the base of the perianth. And what I thought would be cool was that I could take that white area and expand it. And so the next generation, I get that. So I've got pretty much much bigger white area now. And then I can go all the way. This is the one the Dutch are really want. They offered me $40,000 for this bulb. Of course, the Dutch knowing the Dutch, they'll make 10 million. So 40K is, they're going to grow a field of it. Yeah. I'm holding out. This is some of the other jonquilla stuff. I'm trying to get some peachy tones into the jonquillas. This is one of kind of a yellow, white and yellow bicolor jonquilla. I love these that you get so many flowers on a stalk too, which is nice. And they smell heavenly. One of my other things is you need to select for the whole plant. It's not just the damn flower. These things should be good growers. Right now, I'm seeing a lot of selections for dwarfer types of almost everything, partly because I think we're having smaller yards, and I think also it's easy to stack dwarf plants than it is big ones. So one of the things I'm playing with is the hardy geraniums, and I've been trying, which are wonderful garden plants, but I'm trying to make the foliage as exciting as the flowers. And this is a group here where I'm getting them. This one comes up quite yellowish and has brown on it. And the flowers of these are pretty nice too. Here's another one here where it's a more distinct brown pattern, very neatly trimmed. And that's its flower. The next group are nodosums. And I don't know about you, but I don't know if you grow the geranium Roseanne. It blooms forever, but to me, it's the gangly, awful plant. And so the nodosums make nice clumps. So I've been trying to do breed nodosums that have better flower color. And so this next couple are some of the nodosums I've created. And I think these flowers are the equal certainly of Roseanne, but on a clump now. And they also grow in dry shade. All good things. So this is an Oxoniana, sanguinium coeling that I like a lot too. And it has a very good habit as well. And dianthus, they have pollen too. So I'll just run through these. I'm breeding some that are mostly about 6 to 8 inches tall and very, very pretty foliage. Yeah. This is pulmonaria, and so these are miserable things to cross. But what I did is started crossing blues with pinks, and surprisingly, what comes out are blue and pink flowers and striping, and I get it in every combination too. Some quite dark, like that one. Others quite pale. Sometimes it's just the bottom of the flower. Other times it's stripes. And of course, one looks at foliage too. And I've got some rather nice foliage in this one here. And in this one here. I don't know if Nipophias. I was down at a nursery just south of me, and I saw some Nipophias, and I said, oh, I could make them better than that. And so I have. And so these are, I've been trying to get some really brilliant shades. These mango coral shades, I think, are just really good. And boy, they are alive in the garden because you can see them a mile away. You can see that. And then also some of these ones that infuse the green into the yellow. Or you have mango and green and yellow all together. Or white and mango. I know it's annoying, isn't it? Uh-oh. And so the other thing, as I mentioned before, a lot of times we're making shorter copies of things. And so this is one of my aster seedlings here. It's about knee high for me anyway, but it's, as you can see in the background, it's much shorter than its other cousins up there. So what I've done, what I did last year was to cross this particular one with all different colors, and especially some of the very, very dark, because we don't have a lot of very, very dark asters right now. So I took this to everything essentially. And composites are hard to cross because the only cut flowers you use are the ray flowers on the ends, which are female only. And you take your scissors and you chop out the middle, and that's so you don't have those flowers to contaminate. And then you run your pollen right around the outside, and then you collect that seed from the outside. And so you don't get quite as much as the bees do, but you still get quite a few seeds. I've got about 300 seedlings to see this year, and I'm hoping for some good things, but I'll probably need another generation. Oh, I do penstemon too. This is this is one I rather like. I also grade some plants that I was going to do that are very difficult to propagate, and so you have to sell them as strains. But I'm fundamentally against that whole idea, because although that plant is bloomed in my yard, its siblings were not nearly as good, and I want that plant, not the siblings, damn it. Similarly, these are cyclamen seedlings here, and I want the one with a cool pattern. I don't want all the others. And so I'm just going to plant these and enjoy them myself until the Dutch come in and say, oh, we can propagate that one, Kevin, and then I might talk. Some things, Rome wasn't built in a day, and Polly Bishop in 1986 pointed out Xylem Tiny Tim in her garden, and she said, Kevin, it has blue in the eye. And she wouldn't let me leave until I'd made a cross with it, and she sent me the seed. She said, it's going to give you a blue eye. Well, I'm now on the 15th generation from that cross, and I'm getting some blue. So this is one that occurred in generation 9. It has just a pencil blue eye, but I used it heavily because it has sort of a semi-spidery form. And I took it in 2 ways. I took it to spidery things, and I took it to really blue eyes. And it was like magic. I got blue eyes, baby. In all different backgrounds too. And what happens is that there are 3 things going on to make the blue eye: a very green throat, a purple eye, and a watermark. And where the watermark is left, you can see there's that rim of the purple eye still there. And so actually it's kind of cool because now the blue eyes are rimmed with this purple.
Eyeliner.
Eyeliner, yes. Some of them are just gorgeous. And I've got them in various backgrounds, white bases and purple bases. And now here's the group now that went to the spidery shapes. And some very unusual forms came out of this. Yeah. Yeah. So, but all with these tremendous blue eyes. Now, these flowers, the other ones were miniatures, about 3 to 3.5 inches. These are 8.5 inches. So, what a segregation, you know? I like this kind of a multiple eye here. Just some fun things. I do breed some small, very round tetraploids too, and these are just a couple of those. They're such pretty flowers. And I breed small reds, and this is like the one on the screen here looks like it should. The one on the screen up there is a little off. But they're just lovely flowers. Well, I may be a great hybridizer, but I'm the most stupid person in the world in terms of marketing. So luckily I have friends that help me, you know. And so, in fact, Angela from Plants Nouveau is actually in our audience today. They're going to be involved with 6 Louisiana iris hybrids next season. And then I have iris through some of the other usual suspects. And then the sempervivum are sold through either the Chick Charms brand or through Mountain Crest Gardens in California. And here's our heroes celebrating with cake after, after making a deal for Kevin's plants. And so I always found that if you give them lots of sugar, they're bound to make a very good deal. And you can see how happy they all are with their cake. As I said, they're much better at marketing than I am. And so, what Chris has done is to start this new strain of Chick Charm Giants. And so, as I mentioned before, I'm getting things now. I have sempervivum now that are 16 and 18 inches in diameter, because I've been crossing big by big by big now for 3 or 4 generations. And it's because the bees aren't much messing up the works, I can keep getting bigger and bigger. And so my football size semp is not too far off. The neat thing now is that we in the north can grow those big ones, and we can do the same kind of xeric culture that they do in California, using the big sempervivum as kind of in a dry landscape like they do in California. So I think this is going to be very cool. This is the ones that we had a small release of these this year, and then we have another group coming out next year that'll have a few more of these. But they are just, everyone that comes to the yard has been just shocked by these. I had a television crew come in, and I had one in a pot, and he put it next to his head, and it was like twice the size of his head. And so it's like, wow, he was really quite impressed with it. Well, as I said, nothing but pollen is safe in my yard, but I had always hated gladiolas, and then one day Lee Walker happened to cut some stalks of some miniature gladiolas and brought them to an iris meeting. And I looked at him, I said, "Oh, Lee, these are actually pretty nice." And of course, an order went out the next day. They came to my yard, and they had pollen. Actually, I had made such public protestations about that I hated glads that some of my friends actually guerrilla gardened me and planted big gladiolas in my beds. And so I had these things pop up. What in the hell it's like, and finally one of them confessed that they had come into my yard when I wasn't home and planted big glads, just so you know, it would make me angry. Anyway, yeah, I know. Yeah, these are miniature glads. So they're about 2 feet high and have flowers probably 2.5 inches across, and about some of these have quite a few flowers on a stem, maybe 18, 20. I love this kind of these white throats are one of the common things you see in glads. So one of the things I wanted to do is to see what happens when you cross white throats with thumbprint types. And that's what happens. You get these margins now of where the white throat would be, now with a thumbprint inside the. Anyway, they're just the coolest looking things. Yeah. Yeah, that's the other thing I found. I crossed whites into blues, and guess what? They made bluer blues. There's another one here, really brilliant. So, if this wasn't enough to get you to visit, I don't know what is. You can come anytime but Christmas, I think, and see something in bloom. Actually, maybe the heathers bloom at Christmas. I probably, and the hellebores do too. But anyway, so yeah, as I mentioned before, we have the tours coming through Oregon in May, and I'm one of the open gardens, so come see me. Thank you so much for everything.